Hello, this is the online version of Cellular Respiration and Fermentation lecture. Okay, so what is cellular respiration? Okay, respiration is the process of breaking down food molecules into usable energy. It is not breathing, okay, and many students make this mistake. The goal of this form of respiration, okay, is to create ATP molecules. To create ATP molecules and to free up electrons for the electron transport chain because they have high energy. What is this cell respiration we're talking about? It's the process by which living things obtain energy from organic molecules and release the waste products. Primary aim, as we know, is to make ATP. Okay, first we're going to discuss aerobic respiration. Okay, this uses oxygen. Oxygen is consumed and O2 is released. Very important. When glucose is broken down via oxidation, okay, and if we remember oxidation, that's Leo. Okay, a tremendous amount of free energy is released. Okay, so we have two types of cellular respiration. Okay, the first type is aerobic process, which is required oxygen to take place, and a lot of energy is available, so it's very, very efficient. Okay, the other one is anaerobic process, which does not require oxygen to take place, which we get quick energy, but it's inefficient. Okay, so to remember this one, remember AN in science is no, so this means no oxygen or anaerobic. So the deal with cellular respiration is we have to have food. Okay, so when we eat, we get energy in the form of glucose and other sugars, okay, whether it be from celery or a milkshake. That food energy is broken down into usable energy. The energy used to bond phosphate groups, ADP. Okay, so if we remember, that means 2, and to ATP, which is 3. Okay, so we're bonding one more to that ADP to make ATP. We have this glucose metabolism. Okay, this is the chemical formula for cellular respiration. You must memorize this. You need to know it backwards and forwards. Okay, you need to know the numbers, the letters, everything. When glucose is broken down, some of the energy is lost as heat, but much of it is used to make three energy intermediates: ATP, NADH, which we've talked about, and FADH. Okay, we have four metabolic pathways that we'll be going over. The first one's glycolysis. Then we have what's called breakdown of pyruvate. We have citric acid cycle, which is also known as the Krebs cycle. And we have oxidative phosphorylation, which is also known as the electron transport chain. The process of cellular respiration, as seen in your book. The whole process of cellular respiration, all of this portion. Here we have the diagram for it. And so starting off, this involves breaking down one 6-carbon sugar of glucose. So if we see up here, we have glucose, and we can count those little dots, and it's 1, 2, 3, 4, 5, 6. Okay, we want to break down glucose into two pyruvate molecules, which means we're going to have two 3-carbon molecules. Okay, and right there in the pink, we see where it's been split into those two 3-carbon molecules. This process uses two ATP. Okay, so up here in the blue, we see where two ATP are added to the system. It's kind of like a old soda can machine. Okay, you're going to put in 2 quarters to get out a product. Okay, and that's what's happening here. Just think of the ATP as 2 quarters putting into the system first. Okay, this process is anaerobic, which means no oxygen is being used. Okay, and this occurs in the cytoplasm. Okay, so that's an important note to remember, that this occurs in the cytoplasm or the cytosol of the cell. Continuing. So this also creates two free hydrogen ions or free electrons. Okay, these hydrogen ions bond with NAD plus to form NADH, which is those intermediates. Okay, so two NAD plus combined with those two hydrogen electrons, which is right here in the formula. Okay, those NADH then carry the molecules to the electron transport chain. So we're not going to see those again until the very last step. Just keep that in mind. Okay, so we're going to first use those two ATP molecules. Then it converts 4 ADP molecules into 4 ATP. It's happening on each side because there's going to be two products at the end. Okay, so we have ADP, 2 ATP on one side, 2 ATP on the other. We're going to get a net ATP production of 2 ATP for every glucose molecule that we eat. Okay, why is it a net 2? Because we first put 2 into the system to start it, we gain 4, and so we have 2 at the end of the system. Okay, so that's where we've gained 2 ATP. This intermediate step going on. Okay, so in the mitochondria, so we've gone from the cytoplasm into the mitochondria. Okay, we have the pyruvate molecules, which are the 3 compound, is broken down. Okay, so we have that 3 compound molecule coming from glycolysis. Okay, it transforms to carbon dioxide is released, which means 1 carbon is released. Acetic acid, which is a 2 carbon compound, is left over. Okay, so we've gone from 3 carbons to 2 carbons. So that acetic acid 2 carbon is then combined with an enzyme called coenzyme A, as seen here in the green. This forms a compound called acetyl-CoA, which is the whole thing combined, and that is the end of glycolysis. Is we net 2 ATP. We originally gained 4, but because we put 2 in that system, we have an overall 2 ATP gain. And a different way of looking at what I just went over. Okay, to some people, it's helpful to go over a different way and list each step there as well. Diagram greater detail. This is out of the book. Okay, and it goes step by step for you as well to look at. Video that you will need to watch. Okay, you can go to your animations portion to watch this.
Cells
Okay, next we're starting with the citric acid cycle or the Krebs cycle. Middle of cellular respiration. Here's just a view of the Krebs cycle that I wrote in my notes when I was in grad school, just to show you a little uh, clue of how in depth it actually is. We won't be going to it in this in citric acid cycle or Krebs cycle. This produces more ATP and releases more hydrogen ions. The electrons are picked up by NAD and FAD. Okay, and these two are organic carrier molecules. This occurs inside the mitochondria. Okay, that's very important to remember in the mitochondrial matrix. Step 1. So we're coming out of the intermediate step of glycolysis. So the cycle begins with that acetyl-CoA, which is 2 carbons, as seen there in the pink. The acetyl group, or the two carbons, attaches to acetyl oxytate, which is 4 carbons, as seen here in the green. This forms citrate, which is 6 carbons, which is seen in the blue. Okay, so both glucose and citrate contain 6 carbons, but they are not the same molecule. That's important to remember. And this oxaloacetate, the 4 carbon, is already in the system from Krebs cycle. It's a cycle, so it keeps on going and going and going, and so it's already in the cycle there for us. Step 2, that citrate, that 6 carbon molecule, is rearranged to an isomer. Okay, so there we see it in green. The isomer is called isocitrate, and it's still 6 carbons, as seen in the blue. That isocitrate, which is still 6C, 6 carbons, is oxidized. Okay, so there we see that in blue. Okay, this creates alpha ketoglutarate, which is 5 carbons. So that means a carbon has had to go somewhere. Okay, so NAD picks up an electron to become NADH because that carbon is released. Okay, and that CO2, or 1 carbon, is released into the blood. Okay, and so we're going to breathe that out through our lungs. Okay, but that means we're down to 5 carbons now since 1 carbon is left through CO2. Seen there in the step 4. We have that alpha ketoglutarate, which we're down to 5 carbons now, is also oxidized. It combines with more coenzyme A, as seen in the blue. The NAD picks up an electron to become NADH, seen there in the green. And this forms cyclinyl coenzyme A, which is 4 carbons, as seen in the orange. Okay, and it's also going to release another carbon into the blood. So we're down to 4 carbons now. Okay, 2 have gone off into the blood in the form of carbon dioxide, as seen in the purple. 5, we have that succinyl-CoA, which is 4 carbons, is broken down, okay, to coenzyme A, as seen in the blue. Succinate, which is that 4 carbon molecule. This is exergonic reaction, drives synthesis of GTP, okay, which is another form that can make us ATP, as seen in the green there, which can transfer its phosphate to ADP, which gives us 1 ATP. Okay, succinate, which is now that 4 carbon, is once again oxidized. This creates fumarate, which is another 4 carbon molecule. FAD picks up an electron to become FADH from that oxidation, seen there in the green. Fumarate, a 4 carbon molecule, combines with water. Water is already in the body. The combination creates malate, which is still a 4 carbon molecule. Malate, 4 carbon molecule, is once again oxidized, which means it loses electrons. This creates oxaloatate, which is a 4 carbon compound, as seen in the purple. NAD picks up an electron to become NADH from its oxidation, seen in the green. And the cycle is complete and will begin again at step 1 if we remember that oxaloatate combined with acetyl-CoA. Here is that whole cycle again, seen in full view. And so what we need to remember through that entire cycle is we have gained 6 carbon dioxides, 2 ATP, 2 FADH, 6 NADH, and 2 NADH. Okay, now if you're looking at this, you might be saying, hey, the math doesn't add up that you went through in this. Okay, what's important to remember is the Krebs cycle is running side by side as a pair because there were two pyruvate molecules. One cycle creates one ATP, but since two cycles are running side by side simultaneously, it's creating two ATP. So therefore, the citric acid cycle yields two ATP for us to use. Okay, make sure to go to your animations to watch this next video. Okay, here we come to the last step, which is the electron transport chain. Here we're on our last step. Okay, electron transport chain and oxidative phosphorylation. But what's important to remember here is all those NADHs and FADHs that we have been creating, okay, throughout these other two cycles are now coming to this electron transport chain. Step 1A of the electron transport chain, NADA is oxidized to NAD plus, as seen here. Electrons are transferred to NADH dehydrogenase going through, okay, this mitochondrial matrix, as seen in blue. So some energy is harnessed to pump hydrogen ions in the intermediate space, and electrons are transferred to this molecule called ubiquin. Okay, now this might be a little difficult to understand at first, but all those electrons that we've gained in the other two processes are now coming here, okay, and going through this intermembrane space to create ATP. 1B. FADH2 is oxidized to FAD, as seen here. Electrons transferred to succinate reductase, which is another form of an enzyme, seen in the green. This is continued to ubiquitin, seen in the purple. 2, though from the ubiquins, electrons travel to the cytochrome BC1, which is just another protein carrier. Some energy harnessed to pump hydrogens into the intermembrane space, as seen here in the blue, and electrons are transferred to the cytochrome C, seen in the orange. Okay, now if you're following me, you'll see all these hydrogen ions, okay, or electrons over here on this side. Okay, so it's gaining all those electrons that had been brought to us from the other two cycles. Okay, the cytochrome C, electrons are transferred to cytochrome oxidase, another enzyme, seen there in the orange and in the blue. Some energy harnessed to pump the hydrogen ions into the intermembrane space, okay, seen there in the green. And electrons are transferred to oxygen and water is produced. Okay, so as we know, if we add hydrogen to oxygen, we get water, as seen there in purple. Okay, steps 1 through 3 have now produced what's called a hydrogen electrochemical gradient, as seen in the blue. Okay, these hydrogen ions flow down into the matrix through ATP synthase, which is this instrument seen here in green. And the energy within the gradient causes the synthase of ATP to form ADP and phosphate. Okay, so we know when it combines ADP and phosphate, that gives us ATP. So the whole point behind this step is to get those hydrogens in blue down to this ATP synthase to create ATP. It's done with oxidative phosphorylation. So first 3 stages of glucose metabolism yields us 6 carbon dioxides, 4 ATP, 10 ADH, and 2 FADH. These high energy electrons remove from NADH and FADH to make ATP in oxidative phosphorylation. So this typically requires oxygen. So the oxidative process involves the electron transport chain, and this phosphorylation occurs by that ATP. Okay, so this oxidation by the electron transport chain, the protein complexes and small organic molecules embedded in the inner mitochondrial membrane, which we just went over, accept and donate all those electrons in a linear manner in a series of redox reactions. Okay, the electrons originally found in NADH or FADH are transferred to components of the electron transport chain. Each component has an increasingly higher electronegativity, and at the end of the chain is oxygen. So the electron transport chain is also called the respiratory chain because oxygen we breathe is then used in the process. ATP synthase makes that ATP via a process called chemosis. So the lipid bilayer of the inner mitochondrial membrane is relatively impermeable to hydrogen ions. Protons can only pass through ATP synthase. So that ATP synthase harnesses this free energy released as those hydrogen ions flows through it, its membrane embedded region to synthesize ATP from ADP and phosphate. So this process, chemosis, is the chemical synthesis of ATP as a result of pushing all those electrons across that membrane. Okay, so NADH oxidation makes most of the cell's ATP. So the NADH oxidation creates the hydrogen electrochemical gradient used in that ATP synthase. Okay, this can yield us up to 30 to 34 ATP molecules per glucose molecule, but rarely achieve their maximal amount because NADH also used in an anabolic pathways and hydrogen gradient used for other purposes as well. With that ATP synthase, okay, the ATP synthase is a rotary machine The synthesis of ATP involves the mechanical rotation of part of that ATP synthase. Okay, so its conformational changes produces that ATP. Okay, the connections among carbohydrate, protein, and fat metabolism. Besides glucose, other molecules are also used for energy, as we know, carbohydrates, proteins, and fats. These molecules enter into glycolysis or citric acid cycle at different points. Okay, they utilize the same pathways for breakdown as it increases efficiency. I want to watch this overall video once again in animations to understand the process a little bit better, since I can't show you since we're not in class. Okay, you want to watch this one as well?
Proton pumps.
Sorry. And then lastly, we have this anaerobic respiration. Okay, now as I said before, this process requires oxygen, but what if we don't have oxygen in the system? Anaerobic respiration and fermentation. Anaerobic describes the environment that lacks oxygen. There's two strategies to metabolize organic molecules in the absence of oxygen. Okay, they use substance other than oxygen as their final electron acceptor in the electron transport chain, called anaerobic respiration. Okay, so they produced ATP only via what's called a substrate level phosphorylation, or otherwise known as fermentation. So in this anaerobic respiration, okay, we're using here an E. coli uses nitrate as the final electron acceptor under anaerobic conditions. Okay, it also makes ATP via chemosis even under aerobic conditions. Is the breakdown of organic molecules without net oxidation. Okay, so many organisms can only use oxygen as a final electron acceptor. So under anaerobic conditions, they need a different way to produce ATP. Okay, like losing glycolysis. The glycolysis uses up NAD and makes too much NADH under anaerobic conditions, which can be dangerous. Okay, so muscle cells solve this problem by reducing pyruvate into lactate. Okay, yeast solves the problem by making ethanol. Okay, so this is where lactic acid buildup comes from in our muscles. Okay, fermentation produces far less ATP, okay, than oxidative phosphorylation, but since we don't have oxygen, we need to use some sort of. On the left side, okay, we have what's used in our muscles, okay, the lactate. Okay, and as we know, this is painful for those of us that have ever done exercise over and over again and you're sore the next day or you just can't do anymore because of soreness. That's lactic acid buildup. And the other way, ethanol in plants, is the fun way, which makes beer, wine, cheese, yogurt, and all those nice products we like to eat or drink. Summary for the whole unit here. Okay, organisms obtain energy in a process called cellular respiration. The equation for cellular respiration is the opposite of the equation for photosynthesis. Okay, once again, remember this equation. Glycolysis, which is glucose broken down into the cytoplasm through the process of glycolysis. Two molecules of ATP and two molecules of NADH are formed for each molecule of glucose that is broken down. So glycolysis has a net result of 2 ATP and 2 pyruvate. Most of the energy from the glucose is still contained in that pyruvate. The series of reactions in which pyruvate is broken down into carbon dioxide is called the Krebs cycle or citric acid cycle. And the net yield from the Krebs cycle is 6 CO2 molecules, 2 ATP, 8 NADH, and 4 FADH. Transport chain, the final step in the breakdown of glucose, point at which ATP is produced the most, which produces 30. Anaerobic respiration, the anaerobic pathway that follows glycolysis. You have two main types, lactic acid formation and alcohol formation. Remember, this is after glycolysis because it cannot go into Krebs cycle because it doesn't have oxygen. So it goes from glycolysis to these fermentation cycles. Yeah, cellular respiration, we need to know that general formula. Video for completeness. Okay, there's a couple questions here. What's the overall purpose of cellular respiration? Okay, that is to make a present the general sequence of cellular respiration. Okay, that's B, glycolysis, Krebs cycle, electron transport. Cellular respiration is the anaerobic process. That's glycolysis, because remember, if we don't have oxygen continuing, this will then go to fermentation. If we do have oxygen, it'll then go to Krebs cycle. By Krebs cycle is a waste product. Okay, and that is carbon dioxide. Remember, we're breathing it out. It is a waste product. Sure.